Post-mortem changes. So in order to identify presence or absence of cancer, it is very important that histological specimens are fully and well fixed to avoid development of post-mortem changes that would interfere with examination and diagnosis. In histopathology, post-mortem changes refer to degenerative and decompositional alteration in tissue architecture and cellular detail that may occur if the specimens are not preserved immediately. So in case these samples are not fixed immediately, we may have loss of nuclear detail, cytoplasmic breakdown, and bacterial invasion. Therefore, in histopathology, we majorly focus on two postmortem changes. Number one is autolysis, which we call self-digestion of a cell or tissue by its own enzymes, especially lysosomal hydrolytic enzymes after death or removal from the body. When blood supply stops or tissue is removed from the body, oxygen and nutrients are cut off, okay? Number two, ATP production in mitochondria ceases. So, without ATP, okay, ion gradients collapses, okay, and that one will lead to failure of ion pumps, that is sodium potassium ATPase or calcium pumps. Whereas when blood supply is stopped, definitely there will be cell death, which leads to a loss of metabolism. In case the sodium potassium pump is affected, sodium and water rushes into the what? Into the cell, and that leads to cell swelling. And accumulation of calcium inside the cells or the tissues activates destructive enzymes. So number three, in autolysis, plasma membrane and organelle membranes become leaky, especially the lysosomes. So lysosomal enzymes like proteases, nucleases, lipases, glycosidases are released into the cytoplasm, okay? And that increases membrane permeability. Then number four, there is enzymatic breakdown of cellular components. So because of the release of different lysosomal enzymes like proteases digest structural proteins, whereas nucleases degrade DNA and RNA, whereas lipases break down membranes and glycosidases will destroy the polysaccharides. Number five, disintegration of the cell. Complete disintegration of the cell. So, because of the earlier processes, the nucleus undergoes what we call pycnosis, shrinkage. Number two, fragmentation, what we call karyohexis. And number three, we shall have karyolysis, which is dissolution. And additionally, the cytoplasm liquefies, leaving only cellular debris. Maybe you can also look at the effect, what will happen in autolyzed tissues. So once autolysis occurs, number one, there is loss of cellular details because the nuclear structures disappear by karyolysis and karyohexis. The cytoplasm becomes granular, vacuolated, or homogenized, and it makes it difficult to assess malignancy, dysplasia. Autolysis, it leads to poor staining. For example, hematoxylin, which stains the nuclei, fails to bind properly, and that leads to pale or absent nuclear staining. And also the cytoplasmic stains become diffuse. The other effect is misinterpretation. Okay? An autolyzed tissue may mimic necrosis, inflammation or degenerative diseases. And this can lead to what we call false negatives.